So we are here in Stockholm at the Parkinson's Disease and Movement Disorder Society Congress. I just completed the Stanley Fahn Lecture. I was honored to be the 2014 Stanley Fahn Lecturer. This has a great deal of meaning to me because Dr. Fahn is not only one of my closest friends and associates, but he's also one of my mentors, and he's the one who actually inspired me to develop interest in Parkinson's disease and movement disorders. I think mentors are absolutely critical to the development of young scientists and clinicians, and I am glad that I also played a role of a mentor. Sometimes mentors are tormentors, but I hope that I was a mentor to many of my trainees, including Dr. Francisco Cardosa, who is a very prominent neurologist and one of the most prominent movement disorder specialists in Brazil, in Belo Horizonte. So my lecture focused on several topics. One was sensory aspects of movement disorders. I discussed some of the sensory aspects of Tourette syndrome, for example. I highlighted restless leg syndrome. These are examples of abnormal sensory motor integration. So I also described a new syndrome called leg stereotypy disorder, or LSD, which is a very common condition where individuals are frequently restless while they are sitting in meetings or some other situations. This is actually a genetic disorder. It's one of the most common movement disorders, but it has never been studied. So I use this as an example of a disorder of sensory motor integration. I also talked about genetic disorders and highlighted the role of whole exome sequencing in the diagnosis of rare genetic disorders. And then I focused on immunologic disorders, which are becoming increasingly important, and I specifically highlighted anti-NMDA receptor encephalitis, which is often presenting with movement disorder such as stereotypy or facial movement dystonia, chorea, and variety of other movement disorders. This disorder often occurs in children, but can also occur in adults. And finally, I discussed some of the advances in the treatment of Huntington's disease, focusing on VMAT2 inhibitors, which cause dopamine depletion. And I specifically highlighted tetrabenazine, SD809, and an NPI compound, which are the current experimental therapeutics in Huntington's disease. And I concluded with some comments about botulinum toxin, particularly SNARE proteins, which are important in the function of botulinum toxin, and noted that the 2013 Nobel Prize, which was awarded here in Stockholm, was awarded to 3 scientists who developed our understanding of the SNARE proteins and were rewarded for this particular scientific discovery with the Nobel Prize. Thank you. I hope I get invited to Stockholm one day for a Nobel Prize, but it's not going to happen.